And so I'm really going to go into a deep dive into a lot of these different methods in this masterclass. And so you can see some of these topics. So we have quality control, AOI and gene removal, normalization selection, and spatial differential expression. These masterclasses are going to have a simple format of about 30 minutes with 15 minutes for Q&A. And it's really going to cover these topics in general with spatial analysis, as well as how Rosalind is approaching these different topics. This is going to be a quick introduction to our masterclass series. I'm really excited to share with you some intricacies of GeoMx data analysis, as well as how we're adapting to this in our Rosalind platform and our newly released GeoMx pipeline. And what I hope that you will take away from this masterclass is really solidifying the value and synergy of pairing Rosalind and GeoMx at your institution or within your lab. Gain a deep understanding of spatial analysis principles and algorithms and how Rosalind can simplify your spatial analysis experience. Increase your confidence to accurately process and interpret your spatial data and results, and open new avenues for insights that you may have missed or not even considered because you didn't know to look for those avenues. And so I'd like to begin with some background about myself as your masterclass instructor for this GeoMx series. My graduate studies were in translational ischemic stroke research with Nargilin-1 treatment. I have multiple publications in spatial biology and data analysis, and multiple still in review. I was previously a nanostring field application scientist, and so my expertise primarily lies in spatial biology, immunohistochemistry, and neuropathology. I believe that GeoMx is actually a very beautiful technology when we look at something like spatial. It can have its difficulties, but overall it's a really complex, beautiful technology built to intimately and intricately analyze the true essence of what's going on in your biological model. It has three main beautiful steps that you want to pay attention to when you're looking at what's going on in your discovery and in your tissue, no matter what type of tissue you're working with. You can look at the plex and flexibility and precision. So nanostring has, RNA whole transcriptome for human and mouse, but what's unique is you can use this with crossover of any tissue type almost, and still have a pretty high crossover. And their nanostring is unique in their customization. And so you can add custom probes to any panel. And they've now launched with their massive protein panel, which makes me a little jealous that I can't even use it as I'm not a scientist anymore. Which is by far the largest in situ protein panel on the market, which is really cool and still allows customization of additional proteins, which is insane. And of course, super flexible. It's compatible with any tissue type. And the most important step, especially as myself as a brain person, is it's compatible with, your standard histology protocol. And so for really sensitive tissue, brain tissue, bone tissue, breast tissue, it's compatible with your standard histology protocols. You need special slides. You need special adaptations to keep your tissue on the slide for histology. It allows you to make those adaptations and make sure that you're still going to get that data with your tissue. You're working with sensitive clinical bone tissue, breast tissue. You only have one sample. It's going to make sure you get that data. And of course, it was really built for biology. So it's user and tissue adaptive segmentation. Biology is not dots on a slide or squares. It's really unique to every single model, every single tissue, and every single different biology, and everything's going to be very unique to every single thing you put on a slide. And so this unique adaptive segmentation strategy is really built for biology and allowing you to build that research directly to your project in your biology itself. And I think that is really cool. And of course, precision, best informed performance with degraded RNA samples. I was working with a clinician who had 30-year-old FFPE blocks, and they got pretty great data, which was phenomenal. And you can imagine keeping 30-year-old FFPE blocks and imagining you may never get data from that. That's pretty cool. And it's highly reproducible from user to user and site to site, which is obviously a must-have with any data type. And of course, 1 micron resolution. But getting the data and building your project is only half the battle. So what do you do once you have the data? So surprisingly, GeoMx data files are actually really beneficial. They're built really nicely. So once you actually process the FASTQ files from sequencing into DCC files, they're extremely small. They're easy to transfer and store, and most of the data that you need about your project and your different AOIs is within those files. So this allows for rapid data processing and intuitive interpretation, but the difficulties in GeoMx spatial analysis actually lies in the fact that spatial is a fairly new field and it's rapidly evolving. So it's mainly lying in the limitation of software. And so we have limitation. And so the software that we tend to have is very non-user friendly and often requires expert coding knowledge. And so this leads to some downstream issues. So QC can often be inconsistent from user to user. So this leads to, again, inconsistencies. You can have little differences depending on who's doing it, and then also downstream differences as well. It's easy to apply incorrect spatial differential expression methods, and we're going to cover that in this masterclass. This can lead to false positives, misrepresented differentiated genes, blanket thresholding. So these different applications can have lack of transparency, which often leads to loss of low expressors or loss of your key AOIs or ROIs. And essentially you can lose data or be so hesitant to lose these data that you prefer to avoid this step. And again, going back to point one, you can lead to loss, false positives and misrepresented genes. And so all of these items can all lead into each other and cause difficulty in what your data actually may look like. And so we're going to really cover all these items and how Rosalind's approaching these difficulties. And so ultimately, in order to approach spatial analysis, it really requires a depth of knowledge of spatial analysis as well as statistical knowledge, which requires a lot of time. And the spatial field is going to continue to evolve rapidly because it's new, and people are going to continue to learn new things and find new results. And so this requires you to continue to remain up to date with this knowledge and adapt to it, which a lot of these methods can have a lack of adaptability to these new methods. And so a lot of this just continues to feed into each other and make spatial analysis difficult to perform, be consistent, and be confident in your results. And so if you're not familiar with Rosalind, we have many different modules for data analysis that we may cover in certain topics that are available for all of our different assay types. You can see these across our platform. These will continue to be adapted and evolved, such as pathway analysis, biomarker discovery, meta-analysis, and knowledge graphs. But what we're really going to focus on in these next masterclass series is these new modules built specifically for our GeoMx data analysis for WTA, really honing in that Rosalind is the most powerful and collaborative institutional solution for GeoMx data analysis on the market. And so I hope to walk you through each of these items and how Rosalind can approach these different items. And I truly believe that Rosalind will be the best place to analyze your GeoMx data and bring you an end-to-end solution for rapid discovery, bringing you from wet lab and staining your tissue, selecting your ROIs, sequencing your data, rapid discovery within Rosalind, and immediately bringing you back to your next discovery and creating this end-to-end solution for project discoveries on a quick basis.